Okay, I want to talk about where we've been, where we're going, where we are now, and where we're going in a field that we seem to have to measure our progress in terms of decades. I want to emphasise the we in that title there, because I learned a long time ago that if at first you don't succeed, surround yourself with some really smart people and try again, and you'll probably get somewhere. And that seems to be what's happening. So this is just a nod to the team, and I did want to make special mention to Glenn Almany there, who's no longer with us. He was mentioned yesterday, but I can't help feeling that he's here with us in spirit. But I'll move on. Why is quantifying larval dispersal important? Well, there's lots of good reasons. It's the scale at which ecological and evolutionary processes operate, scale at which populations respond to and recover from disturbance, scale at which fisheries, I suppose, should be modelled and managed, although it's really worth pointing out that no fishery that I know of has ever really been managed on the basis of real information on larval dispersal, and also the scale at which marine systems could be designed to meet certain goals, such as larval export and recruitment benefit and so on. But I must admit, these are the kind of reasons that I trot out in the introductions to my papers, and I have to confess the real reason is that this is just a big mystery, and or has been in marine biology, and that's especially true for coral reefs. So it's quite easy to go out on a coral reef and watch fish spawning, and that you basically see the spawn, and then the next generation just disappears in a puff of gametes. So you can't help wondering, where are these things going? And you might go out on the same reef a bit later, you look at the little juveniles of all the different species turning up, you can see them on the day they settle, but they just kind of arrive there, it's manna from heaven. You've really got no idea where these things are dropping in from, where they've come from. So that's really what has concerned our research team. And over the past 15, 20, 25 years, I think we have gone a long way into unraveling some of these secrets in terms of where they go. But there's still much to do, and I think the best is yet to come, actually. So I think it's fair to say that pre-1990s were the dark ages, because I was pretty much there fumbling around in the dark, and there was this tacit assumption of long-distance dispersal. Populations were assumed to be completely open, things would never come back to where they started from. Populations were connected over hundreds of kilometres, and the evidence seemed overwhelming. There was long pelagic larval durations, modelling work, and so on, but most of this stuff was indirect. It was a strong belief that underscored most of the papers in the first book. There was a collection of papers on the ecology of reef fishes. The only thing that I'm thinking about in hindsight is how firmly we held that belief in the absence of any concrete data on where larvae go. There just wasn't any information on that. 1991 to 2000 was a really exciting decade of discovery for me. There were lots of studies coming out using a range of different techniques, population genetics, otolith chemistry, biophysical modelling, and the first attempts to actually tag larvae and see where they go. And a lot of these new approaches were starting to detect or suggest strongly that self-recruitment is happening in coral reef fish populations, and that idea just started to emerge by the time the second book on coral reef fish ecology came along. My own particular contribution to that back in the 90s was larval tagging using fluorescent compounds, tetracycline, so we could immerse embryos that we would pick up off the bottom, put them in a plastic bag and subject them to tetracycline, which would then etch a fluorescent core on the otoliths of these juveniles. So we went out at Lizard Island, we tagged millions of embryos by doing this to them. We collected over 5,000 juveniles that were turning up at Lizard Island, and we just had the simple question, were any of these coming from Lizard Island? And we found 15. 15 doesn't seem like a lot, but we only tag such a really, really tiny percentage of the population. So even if Lizard Island was totally self-recruiting, we would only have expected 50. So there's 30% self-recruitment. But it just kind of begged the question, where are the rest of them coming from? How far have they travelled? 2001, 2010 really was the decade of connectivity. This was a period where there was just a huge amount of research effort that started to be directed towards trying to figure out larval dispersal patterns. You'll see there are a whole range of different techniques that were being applied, some good, some bad, but we learned an awful lot about dispersal during that period, and much of that can be found in the latest book on reef fishes, which has actually just come out this year. But over that decade, well, what did we find? Some self-recruitment turned out to be the rule, not the exception. A lot of different studies there on a lot of different fish. That's percent self-recruitment, so that's the proportion of juveniles that arrive at a population that came from that source or natal population. And it's never 100%, it's never zero, okay? There's a whole range of values, maybe the average is 30, 40, 50% or so on. So again, it just kind of begged the question, okay, we know what's happening now with a certain proportion of the production, but, you know, what about the rest of the dispersal curve? So particular highlights for our group during that period was to develop a method of genetic parentage analysis to try and get at dispersal. And this is really a DNA approach where you sample juveniles and essentially just match the DNA to the parents. Proof of concept, obviously, there with clownfish. But this is a very powerful approach because it provides individual dispersal vectors, you know, where the parents are, you find the offspring, you know, you can get an exact dispersal vector. So this enabled us to look at self-recruitment, connectivity across multiple generations for the first time, and to maybe validate some of the other methods for looking at larval dispersal. So during that decade, we took this technique to Kimby Bay and to a little island where we found a fairly isolated population of a clownfish called the Panda clownfish. And we were able to find juveniles coming back into this very small population. So this was the smallest scale of dispersal that had been documented for a marine fish. That area is about 300 metres across. And we even had some juveniles coming back to their natal anemones. So that happens. The other highlight for us, we developed a new technique for marking larvae during that period. This is the maternal transmission of stable isotopes. So you basically take an injection of a trace amount of stable isotopes of elements that are found in bones. The mother passes this on to through the eggs to all of the offspring, and the core of the otoliths that develop, and all the juveniles have a very distinct mark because that's the material that's come from the mother. We then took these two techniques to another little island called Kimby Island, which is now pretty much home away from home for us, where we've been combining parentage analysis, stable isotopes. Looking here at two species, the true Nemo and vagabond butterflyfish. There's two species with completely different life histories, one with a short larval duration, one with a long larval duration. But surprise to us, we found that a large proportion of the recruitment at that island is coming from adults on that island using both of these two techniques that give us fairly similar results. It wasn't really till the end of the decade that we actually managed to track larvae going from one place to another. And then we did this by moving around to the adjacent islands and really, you know, just sampling lots and lots of juveniles and really looking for tagged or individuals that we could assign to parents out at Kimby Island. So that had happened. But we still felt a little bit dissatisfied with this. By 2010, no dispersal kernel had ever really been fully described by any there's no really one technique that you could use to really get at this problem. biophysical modelling, otolith chemistry, population genetics. They're actually quite good at telling us the large-scale potential for where things can get to. But when it came down to sort of looking at individual islands or relationships between local populations, they just don't have the precision. So that was the problems for scaling down. For us, larval tagging and genetic parentage, it told us a lot about self-recruitment. But its limitations seem to be this is really just going to be a local thing. We're not really going to be able to scale this up to sort of describe the whole dispersal kernel, but we set out, that was our challenge, to go and do that. And if we couldn't, at least to be able to cross-validate our approach with some of the other techniques. So what do? We started with our beloved Kimby Island, and then the basic thing was to just expand the study. And so we focused on these other little islands around the bay, and we just moved around the bay sampling the populations, trying to sample as close to 100% of the clownfish as we could at all of these places. And these places weren't just chosen randomly. They're places that are super concentrations or high densities of these species. They were also part of a marine zone network, so they're all at least on paper, some sort of marine protected areas. And look, this is what we found. This is what the populations look like in Kimby Bay. It really is a large meta-population. You'll see there that there's self-recruitment pretty much happening at every single island that we looked at. But we're also getting a lot of connectivity, and the connectivity is bidirectional. So pretty much every population is connected to every other population in terms of dispersal. These maps are based on sampling tens of thousands of individuals and doing the DNA work on them. And I just want to highlight these ones. They went in both directions across the bay. That's 110 kilometres. These little babies are moving 10 kilometres a day out in open water. And they're doing that in both directions. And we feel that that's about the limit to what these things can do in that area. This hides a huge amount of effort for which I owe Glenn Ormany a great deal of thanks. Because he was involved with all the inshore stuff of this. There were just so many volunteers associated with this. But 2009, 2011, very similar pattern. So the story is self-recruitment at all these islands, connectivity almost between every single other island, and this is running unexpectedly in both directions. So where we are now, this is really for us, I think, the first full description of a dispersal kernel for a marine fish. We're very confident in the shape of the dispersal patterns for this region. It's an exponential decline in dispersal with distance. Actually, the mean observed dispersal distance was 10 to 15 kilometres only. When we model that to take into account of the population sizes and the reef spacings and so on, it's very similar. 95% of the recruitment is less than 40 kilometres for the species. They can go 100 kilometres, but it doesn't happen very often. Incidentally, the average mean inter-reef distance is 62 kilometres, so they're falling out way short of what you'd expect on the basis of just the spacing of the reefs and consistent patterns over 2 years. What about the butterflyfish with the really long pelagic larval duration of 25 to 48 days? Look, it's still a declining function with distance, but it clearly goes a lot further than what we were able to sample in Kimby Bay. The modelled mean dispersal distance for this species is more like 150 kilometres, okay? But you do get similar patterns of connectivity on the scale within the bay of multi-directional connectivity and so on over the same spatial scale. But this time, 95% of the recruitment is likely to be less than 500 kilometres. So they go a long way. And we've been able to develop this technique not just for these small reef fish, but for larger reef fish. This is the work done down on the southern end of the Great Barrier Reef that we did with Hugo and so on. This is the inshore coral trout species where we targeted green zones. We were interested in whether or not green zones are exporting juveniles to fished areas, and we applied the parentage analysis to do that, and we were able to show that they do make a pretty significant contribution to the areas. The reproduction is not locked up in the reserves, it is being spread around at that scale. And the latest on that is that we've been able to sample over a much larger scale, up to 250 kilometres. You can see there the Keppel Islands in the middle, we've got the Percy Islands to the north, and the Capricorn Bunker Reefs. So the same story is coming out, this huge range of dispersal patterns. Everything is not just pushed downstream by the current. We get local recruitment, but we do get very long distance dispersal. And in this case, that movement in both directions between Percy Islands and the Capricorn Bunkers is the longest distance that we've actually managed to track a marine fish larvae in the oceans. And again, the story is both directions, and obviously a lot of mixing between all the different reef zones, management zones, and so on the reef. So this has been really exciting work for us to do. Where we're going, we really want to understand the ecological and evolutionary significance of the two ends of this dispersal kernel, assuming it's some sort of declining function, understand the importance of self-recruitment and that long-distance dispersal, cross-validate the dispersal kernels with other approaches. So we're doing a lot of work with biophysical modelers and so on to see if this all matches up and we're getting the right kinds of stories, and to come up with some models to look at how reserves or populations are working based on real dispersal data, which really hadn't been done before. Just a little bit on that. We have some progress. We're using DNA now as a demographic tool, so we sample a population every 2 years. We take the DNA samples with little fin clips of every single individual on the population every 2 years. This is Kimby Island, and we know that this is sustaining itself over a long period through consistent self-recruitment plus immigration. We can, for the first time, sort of look at the internal dynamics of just one population, which in itself is made up of small units, subpopulation units, some of which are self-recruiting. And we get very real consistent patterns with certain lagoons accounting for most of the self-recruitment that's going on in the island. And we've been able to do this. Oh, sorry, this is just the stage-specific demographic model for that island. And the conclusion from these figures is that this population can sustain itself on the basis of self-recruitment. There is enough self-recruitment happening there to keep that population going into the future. Individual parts of the population can't, but the island as a whole can. It doesn't need the immigration. But if, for example, this population was to go extinct, that immigration would be enough to rescue the population and allow it to build up again. You can see what I mean. So this is a very sensible life history for these fish, and it means that population, to me, suggests there's a certain amount of resilience to that population. Look, this is the other thing that we've got for the first time. I didn't actually ever think this would happen in my lifetime, but we're getting now the first family trees for marine fish I really didn't think this was going to be possible. So don't worry about the details there, but that's just one family tree that's going on at Kimby Island, where one of the longer one was for 5 generations, and we have that just in the 10 years of sampling. So this is coming out through juveniles coming back to the same island, breeding there, and their offspring coming back to the same island. They don't all come back, obviously, some go somewhere else, but a certain proportion are coming back. Occasionally that pink square actually means that they actually come back to their natal and end up breeding with related individuals. This is rare, but it does happen in this population because it's relatively small. But anyway, 5 generations in 10 years, it really suggesting to me we're going to be able to in the future get at evolutionary questions, local adaptation questions in these species, and we do have a little limited amount of evidence to suggest that the ones that come back to Kimby Island actually perform better than the immigrants. But I need to wait on that. I just want to show you this. We have a very good 3D biophysical model for Kimby Bay because it was actually difficult to understand how we could get all this multi-directionality going on in this system. But you'll see there, these are releases from opposite sides of the bay, and you can see they spread in both directions from those points. It's a very complex circulation which accounts for the fact that we are getting all this bidirectional dispersal happening over distance. So the model, the parentage data is actually giving us a pretty good match to this model. It was developed at Woods Hole. It's accounting for self-recruitment at isolated islands, okay? It does require a good swimming ability and a good ability of these things to re-find the reefs that they're at. And the other thing we're finding is that big populations in the inner part of the bay are very important and contribute. They're retained more across the whole bay, but spread across a bigger area. Just finishing, the other work that we're doing, and this was introduced yesterday by Sean, trying to use this sort of real empirical data to develop dispersal kernels that we would then apply to look at things like how marine protected areas are working. You know, are they benefiting outside? Are they benefiting populations in the long term for conservation? I won't say too much about this, but it does really support the argument. Well, reserves are certainly protecting populations between generations within reserves, and we're getting that build-up of populations through recruitment. The blue lines on there are the fished areas, and you can see that after a bit of a lag phase, the reserves, as they build up and start supplying juveniles to the adjacent areas, you can see that really are adding to the fishery yield. There's a little bit of an initial dip through displaced fishing effort, but eventually the contribution or the recruitment effect from those reserves can be seen. The real exciting thing to me about all this is the fact that we're now doing this for real populations setups and using the dispersal data. So emerging generalizations. Dispersal kernels seem to be a declining function with distance for pretty much everything that we've seen. Obviously very steep for the clownfish, flatter for things that have a longer PLD, but they're still declining functions. Dispersal kernels broadly overlapping, but obviously long PLDs or pelagic larval durations mean a long tail, and perhaps that old idea that things are being dispersed down current hundreds of kilometres definitely could be applying to those species. Both self-recruitment, multi-directional connectivity appear to be a feature of population persistence, perhaps much more so than we would have said 10, 20 years ago for these kinds of species. Just to finish up, it's worth, I think, speculating a bit on where we might be going to go in the next decade. I think dispersal kernels will end up being fairly routinely described using parentage analysis. I think there was a time when people thought it was just a boutique thing that you would apply to a few specialised species. But I think now the count is up to about 12 species for which this has been used, and they're refinding juveniles for some species with quite large pelagic larval durations. We'll have a bit better understanding of the ecological evolutionary significance of local retention and long-distance dispersal. We're putting a lot of effort into this, and we'll obviously move further on the trying to understand how marine reserves work in relation to these dispersal patterns. And there finally, I think, we're putting a lot of effort into just figuring out how these fish out in open water find their way between reefs, and so we'll have a better understanding, I think, of how Nemo finds his way home. So thank you. I'll just